Hello, I'm Ian Flinn from the Sarah Cannon Research Institute in Nashville, Tennessee. I'm joined today by Loretta Nastoupil from the MD Anderson Cancer Center and Amitkumar Mehta from the University of Alabama at Birmingham. We're going to talk about some of the exciting news that's come out of ASH 2021. It was a big year for lymphoma studies this year, and I think we'll have a great discussion. So Loretta, we've seen a number of new studies on follicular lymphoma also at ASH 2021 this year. One of them was the Mosunetuzumab phase 1/2 trial in patients with follicular lymphoma. I think, one of the thoughts is that perhaps these bispecific antibodies may displace CAR T cells, or at least give CAR T cells a run for their money across a broad array of lymphomas. Here we have, I think, fairly impressive data with Mosunetuzumab in follicular lymphoma. Can you walk us through that trial and the data?
Sure. So Mosunetuzumab is a CD20, CD3 bispecific antibody, and one of its advantages is that it has a longer half-life than, say, blinatumomab, and so it allows for less frequent dosing. It also has a sub-Q formulation that's under development. What was presented at ASH this year was the expansion study in relapsed refractory follicular lymphoma. These were all patients in third line or later. They had all had a prior alkylator and CD20 antibody. They do receive a step-up dosing because CRS is a common side effect we see across all these bispecifics, and maybe the rates of grade 2 or higher might distinguish one from another, but they get a step up, meaning a low dose on day 1, a little bit higher dose on day 8, and then finally full dose by day 15, and then they're dosed every 3 weeks beyond to either up to 17 cycles, so there's a fixed duration of treatment with this agent as well, or if they're in a CR, they can stop after 8 cycles. And so again, in a pretty heavily pretreated patient population where other therapies that have been approved in the setting result in a median PFS of around 12 months, we see a median PFS of about 17 months in this patient population, and in a good portion of patients that only had 8 cycles of treatment, suggesting that the durability also is encouraging. So I do think that this will challenge CAR T and follicular lymphoma, and I think this offers an effective strategy for patients. And I think the next wave is now to move it into earlier lines and in combination.
Perfect. So, Amit, what about this for the average community physician, giving therapies for patients with follicular lymphoma? I mean, they have the PI3 kinase inhibitors, they have a variety of different options for patients. If this gets approved, where do you see it fitting into that line of therapy? And I guess also just the administration of it, if it's intravenous, and the toxicity profile.
So bispecific antibodies, they are making their headway. Actually, as Loretta mentioned, impressive activity in POT24 and highly refractory, double refractory follicular lymphoma. And if you look at third line plus therapy in follicular lymphoma, maybe you're comparing with PI3 kinase inhibitor. They're oral, so they have added advantage being an oral therapy compared to the bispecific antibodies. But the response rate and tolerability of bispecific antibodies, they're way better than the PI3 kinase inhibitors. Also, there are multiple combinations being explored, including with lenalidomide as well as polatuzumab, and all data were presented at ASH, a very impressive activity, including not only just mosunetuzumab, but glofitamab. Also, both of them, they're being explored as a subcutaneous injection, like we know epcoritamab as a subcutaneous injection. So I think CAR T has done one important thing is to recognize and treat CRS and ICANS. So academic centers, they're very equipped to kind of identify those specialized side effects and treat them. I think community, it will take a little while to kind of adapt to that. And importantly, we all know that, these patients are being admitted in the hospital for a brief period of time to kind of observe. And if they develop CRS, they need tocilizumab or steroids. I'm not sure whether the community doctors would be able to have that facility with an expert team available on their hands to kind of deal with those. PI3 kinase, as you know, that there are 4 different kind of PI3 kinase already approved in follicular lymphoma setting, but they still are very active and they have a peculiar side effect in that setting. Compared to bispecific, the response rate are relatively lower, and uptake in the community is also lower because, right from the beginning, diarrhea, transaminitis were challenging side effect, including infections in this particular group of agents.
So Loretta, do you think that this requirement for hospitalization or the need for having tocilizumab or all this other, all these issues with cytokine release are permanent? Do you think that when we know that some trials with some of the bispecifics don't necessarily require someone to be hospitalized? I mean, that could be a huge speed bump for community physicians in using this type of therapy.
Yeah, I think it depends a little bit on which agent. So for instance, Mosunetuzumab, it's no longer required for them to be preemptively hospitalized, and the rates of grade 2 or higher CRS are exceedingly low. So that's a little bit different than say glofitamab, which we saw some data, but I think what all of the companies are doing, and probably why it's delayed the approval in some sense, is trying to figure out mitigating strategies, because that will be a deal breaker. To have to hospitalize someone just to observe them for potential toxicity is not really feasible, and then it's not really going to replace CAR T, in my opinion. But if you could identify patients who are at risk and employ some of these strategies, so what they are looking at is more aggressive pre-meds, so 20 milligrams of dexamethasone, for instance, the step-up dosing. Glofitamab is utilizing obinutuzumab to essentially compete with antigen binding sites, so you have less supraphysiologic activation of T cells during those first few weeks, and then the sub-Q administration. I think the other thing that we all need to do is, as we gain more experience, be able to identify those patients who are at higher risk, which I'm sure it's going to be somewhat dependent on tumor burden and whether or not they have circulating malignant B cells. If we can do that and identify those that are never going to need TOCI or hospitalization, yes, I think it can be done safely.
Perfect. Thank you.